So I'm just going to touch briefly on the Gtex project and insights from that I think can be very relevant and very important for the Human Cell Atlas. And I'm going to cover some of the design of the project, including its donor recruitment and the various standards it put in place. I'm then going to give you some of the latest analysis insights we've gleaned from Gtex and the latest data release, and how those play into what we've learned or what we need to learn with respect to cell composition, and then briefly where we are and what we need next. So for those of you who are unfamiliar with the Gtex project, although it sounds like from my introduction everyone ought to be familiar with it, but for those of you who are not familiar with it, it stands for Genotype Tissue Expression Project. And it came on the heels of the disease-associated GWAS variants that were discovered for all the complex diseases, so right after the GWAS era. And the observation that the vast majority of those associated variants are non-coding. So that came with the hypothesis that the functional effect of those non-coding variants is through changes in gene expression. And we can measure those regulatory changes in gene expression through this technique called expression quantitative trait loci that you heard about this morning. And that's shown here. We need to measure them across different tissues because diseases are tissue specific and because gene regulation is tissue specific. So here in this cartoon, you can see in the lung, we have a local genetic variant that affects differential expression in that tissue, and we see this classical correlation of expression with genotype. We don't see that in the heart, and there we don't see a correlation with expression and genotype. So to assess the role of genetic variation on gene function requires assessing it in two dimensions. You really need to look across populations of individuals so you can get this genetic variation and the effects of genetic variation, but you also need to look across multiple tissues and cells within individuals. And so that was what the Gtex project set out to do. I like to think of the Gtex project as the Human Cell Atlas before we got to the cell part, so that would make it the Human Tissue Atlas or the HTA, but I think that's now used for a Human Tumor Atlas perhaps. But in any case, one of the things Gtex set out to do was the same as the Human Cell Atlas. That is to create an atlas of gene expression across human tissues. In addition, it looked at this genetic component, so it was looking at the expression regulatory variation or EQTLs. So we needed to sample across the human body and across multiple individuals, and the project sampled about 54 non-diseased human tissues. Again, like the Human Cell Atlas, we wanted a normal, non-diseased reference, which is pretty hard to get for tissues, as many of you have discussed today. And we wanted to do it across multiple individuals. So there were 54 tissues collected with 11 distinct brain regions, and the core molecular assays then to get at this were to have a genotype across everyone, so we had a whole genome sequence performed, and then to do RNA sequencing data to measure the transcriptome across all the tissues. In addition, we knew that to understand the functional consequences of genetic variation, we needed to look at other molecular assays, including proteins and epigenetic assays. So there are a suite of additional assays performed, including protein quantification, DNA methylation, histone modification, open chromatin assays, and so on. And these are just starting to be published. We have a suite of Gtex papers that have just come out as preprints. And more of them will come out soon. These were not done as systematically, however, so they weren't done across all the suite of tissues by donor axes. So what are the key features of the Gtex pipeline then that I think can pertain to some of the things that we've discussed today and that we're doing in the Human Cell Atlas? And these are them in a nutshell. One is we managed to survey a large number of individuals. So the largest study we saw today was the lovely blood EQTL study. Gtex is about that same size, which is pretty impressive to do across tissues. So a large diversity of donors and multiple normal tissues, so non-diseased tissues across the same individual. So these obviously had to come from deceased donor sources to collect that many tissues. It's extremely hard to get tissue, at least across the body in this way, from living sources. So we had a network of deceased donors from organ transplant donors and rapid autopsy postmortem donors. The quality of these tissues obviously had to be extremely good, so we had to get very low ischemic times with collection teams. And for all of these donors, we enrolled them using a consent mechanism where we consented them through their next of kin. We also engaged many of the donors in the study for ethical reasons, to understand what were the reasons they were not participating in the study or what reasons they were. So it had a very big donor engagement process. One of the things we needed to do is you really want to sample the same tissue in the same way across the multiple donors. So we needed accuracy of the site of sampling and coordinate references. And we needed consistency and repeatability, so rigorous sampling and training and SOPs at the sites. In addition, we collected an extensive amount of metadata and clinical data, including phenotypes, medical, histology, pathology, and additional collection covariates and analytical metadata, which are all become very, very valuable covariates in analysis. So this is what the donor collection pipeline looked like. As I said, we had two types, transplant organ donors from OPO sources and post-mortem donors from rapid autopsy sources. And you can see those differ in a couple of critical ways here, which I can't read that well myself. But the OPO donors were largely either had brain dead or significant brain damage. They were all on ventilators. We could have a collection team ready in the operating room following those because there were collection teams ready to do the organ transplant, and then they went in immediately to collect the Gtex tissues. In contrast, in the post-mortem donors, those were mostly accident or trauma victims. They were only occasionally on ventilators. The collection teams weren't ready in the OR, but they had to be assembled as quickly as possible to get there to do the collection time. So the transplant donors obviously had the lowest ischemic times. The post-mortem donors had longer ones, but it was variable, and many of them were still acceptable. Over the study overall, as I said, we used next of kin consent. All racial, ethnic groups and sex were acceptable into the study, and we tried to obtain all of those. It was an adults-only study just to limit the variation because of the numbers of donors. And brains were collected, but only if they were not on ventilators, so that meant most of the brains came from the post-mortem side. And this is what the collections looked like. You can see they were set up very rigorously. This is teams in the OR. All the 54 tissue tubes were set out, pre-labeled, very organized where they were going. These teams were trained. There's the cassettes that the tissues went in that are all labeled. I've already mentioned this was a normal non-disease reference population, and what we really had to put in place was rigorous repeatable sampling, because we needed these teams to sample the same tissue in the same way at the same site as much as possible. So at the point we did Gtex, we did not use the term common coordinate framework, but that's essentially what Gtex laid out through this use of standard operating procedures and descriptions of where to sample and the training of the sites that did it. And this is what those SOPs looked like. This is the tissue harvesting work instruction. These are all available on the Gtex website and in the NCI's website, so they can be reused, obviously, or adapted for collections used in the HCA. This one is on page 2 of 21. They were very detailed about how to prepare the aliquots, what the organ priority was, what location it was. And that's because we were collecting on average about 45 different tissues per donor with multiple different aliquots, some being fixed, some being frozen, and so on. So these SOPs describe the precise size and preparation of the aliquots. And you can see this picture here of the lung where we wanted to get that upper left lobe and that cross section, take that out, and then the size of the tissue. So this is the kind of detail that went into it. And this is just a couple more pages showing other organs. Many of them had detailed text descriptions. Some of them had example pictures as well, such as the right atrium here and the gastroesophageal junction. And this goes on for all of the different tissues and organs that were collected. In addition, one of the key things was to preserve a sample for histology and to get a pathology review of every single specimen. So you can see in the picture here, they have the tissue. They're cutting it according to spec and size to go into the different vials based on the sort of schema that we laid out, where an adjacent piece was to go for histology and one for molecular analysis. H&E slides were made of those tissues, multiple sections of them, and then those were all digitized for pathology review. And there are, again, as I say, 2 places you can actually go and zoom into all of these images on the Gtex portal, as well as the NCI site if you're interested. And they're digitizing those and making those available, and if that's done in the HCA, in the DCP as well, allows you to have multiple independent pathologists conduct review and see different things of these. So despite that rigid SOPs, you still get tissue sampling heterogeneity, and that's shown here for 3 tissues: breast, the heart, and lung. So you can see going from left to right, on the left are generally our better quality ones, and these are all the annotated pathology comments that the pathologists added to these samples. So for breast, you can see on the top left, we have a sample that's predominantly fibrous stroma with many ducts and lobules. On the right, a sample that's predominantly fat. Basically. For the heart sample, we have pieces with no abnormalities on the left. As you move to the right, we see extensive scarring and fibrosis, suggesting an old infarct, large vessels, nerves, and fat. And the same for the lung. On the left, a piece with minimal congestion that looks very good. On the right, 2 pieces with moderate to marked acute chronic pneumocystis and congestion. Now, what the pathology review did is basically to leave all of these in but annotate them. So they rejected any tissue that was grossly abnormal, but this kind of variation they left in the study because this was considered normal for age, or for the age and the state of the individual, health of the individual. So this normal range varies a lot. It varies a little bit by how they were collected, and as many of the tissue atlases for the Human Cell Atlas have reported on in the main meetings, this is the kind of thing we expect because there is no normal region, even with a rigid sampling SOP. A heart varies very much from individual to individual, as is the case with most organs. But it's important to have this variation and to document it because it's very correlated with the changes in the transcriptome. What we see here is Gtex stomach samples, and the first 2, they're plotted on this PC plot for PC1 and 2, and you can see that they fall into 2 distinct clusters. When we went back and looked at the histology images for those, we found that there were 2 sort of extreme types of sampling for the stomach. One on the right has a good mucosal layer. The one on the left has lost that layer and it's largely muscularis. So we performed a laser captured dissection study where we took those 2 sections from the tissue, took the markers from those, fed it back into this study, and lo and behold, these 2 clusters correlate almost entirely with the sampling heterogeneity for those 2 samples. In addition, we see donor collection correlates with RNA quality and hence with downstream transcriptome as well. So what I'm showing you here is simply just the RNA RIN score or the RNA quality score for those 2 different post-mortem donor collection types. On the right are those OPO donors where I said we have people in the operating rooms ready and with the lowest ischemic times, and you can see each one of these is a donor on the bottom. The box plots represent the quality across all of the tissues that were sampled for them, and you can see that the RNA quality in those tissues is almost uniformly very high. On the left are these post-mortem donors, and you can see that it's far more variable. There's a handful of donors where the quality overall just fell below, and these ended up never making it into the Gtex study for RNA sequencing. There's others where the quality is almost as good as the OPO donors, and then there's some where some of the tissues are quite good quality and some are less. So this donor, even at the very front end of the collection, affects the downstream analysis. So capturing extensive metadata is very important, and Gtex captured a lot of it. So what I'm showing here are PC plots across all Gtex donors for, again, those 3 tissues. It won't surprise you that sex is strongly correlated with a difference in the transcriptome for breast tissue. But that's not true. Sex isn't one of the major drivers or the most strongly correlated covariate for the other tissues. For the heart, left ventricle, it turned out to be ischemic time. That's quite strongly correlated with the expression differences. And for lung, it's the cause of death. You can see one cluster here on the left that is very distinct from the other clusters. And I'll come back to that in a couple of slides. So this then is the Gtex V8 data release. It represents 17,382 different tissues across just shy of 900 donors. You can see that we don't have every tissue. This is just a little plot across the tissues with the donors on the y-axis. We don't have every tissue represented for every donor, and that's for multiple reasons. As I told you, we don't get all tissues collected across all donors, so the brain were only collected on donors that weren't ventilated. Also, there are some donors that are organ donors where there are organs that end up going for transplant, such as kidneys, lungs, hearts, so we tended to get those less frequently. And then not all the tissues yielded RNA of sufficient quality, and that was variable by the tissue type and by the type of collection up front as well, as I showed you. So our core data types then were really the genotype and the transcriptome expression, and what we produced from that were expression quantitative trait loci and splice quantitative trait loci. So those are the core data types that comprise all the analyses that have been released on that preprint that's just out. And from those, we looked at variants in cis to generate cis-eQTLs and cis-splicing QTLs. But the data set was now large enough that we could also do analyses of trans, so we looked at trans-eQTLs and trans-splicing QTLs as well. And basically what we find is that the genetic effects on transcription are widespread. So every dot here is one of the Gtex tissues colored by our color coding system. They're sorted by sample size. For some, we had more samples than others. And this is the number of E genes we find. And basically, we find evidence of regulation by local genetic variation pretty much for every gene at this point. As we heard this morning, we're now well-powered enough to discover independent, multiple conditionally independent signals for many of these genes as well. So these are the tissues on the bottom sorted in by sample size going right, and you can see that there are many tissues there that have 4 or more independent eQTLs per gene, and the red dots show the mean of that, so a mean of about 1.8 to 2 independent eQTLs per gene. So over 97% of protein coding genes and about 80% of protein coding genes have at least more than one eQTL and more than one splice eQTL respectively. And this dependency on sample size suggests that we'll probably discover even more loci if we can go beyond these sample sizes to larger numbers. So there's one question here: are these additive and independent effects, or does this reflect the tissue the cellular composition of these tissues? I'm not actually going to go into that data, but I'll tell you it definitely does reflect some of the latter and probably both. One of the things we want to do is actually to fine map these QTLs, because our goal really is to get at what's the causal variant here. And so we did that. The data set's large enough. We did that with multiple different techniques, and we're still doing it with collaborators using a couple of different further assays. And what's shown here are E genes with variants that are fine mapped with greater than 0.5 posterior probability of causality. So a high likelihood of causality. You can see that the methods didn't differ very much. On the top are the eQTLs with basically strong effect sizes, allelic fold change greater than 1. And you can see that we're pretty good at detecting those, the causal alleles for those, across the spectrum of sample size. But for variants with lower allelic and weak effect sizes, you can see that we're very sample dependent there. So we need much larger sample sizes to detect causal variants when they have a low effect size. On the right is just one of the vignettes from the paper that we have in the bioarchive, where you can see several red dots. So there were several eQTL variants in very high LD, and we're able to use these fine mapping approaches to map down to one that lands very highly in a binding motif of a ChIP-seq peak. So in terms of specificity and sharing of these eQTLs, in general, there's a lot of sharing, as you can see. This map is pretty red overall, but there are blocks. Down here, we have a brain block. At the very top, we have the skins together, some muscles, a large group of tissues sharing in the middle. And then we also have up the end here some tissues that don't have a lot of sharing with others, including testis samples and blood. That's one of the things I'll mention about blood. Blood studies are going to forge ahead in the single-cell world for EQTL studies because they're so accessible, but blood isn't always a good proxy for these other tissues, so it is important for us to do these tissue studies. Down the bottom here, we see the different data types, and you can see there's a lot of sharing across the QTL types, with the trans possibly having a little less sharing than the other data types. But in general, what we see for both splicing and expression QTLs is that there's either this U-shaped curve where these are either very tissue specific, they're only present in one tissue, or they're very strongly ubiquitously shared across the tissue types. So the question then is this specificity and sharing driven by regulatory activity that's tissue specific or tissue shared at the level of cell types? And to answer this question, we really need this cell type specific EQTLs, either single cell data or or the way we approached it, to use a deconvolution approach computationally to deconvolute cell types in the Gtex data. So we applied a computational approach to map what we call cell type interaction QTLs. These interaction QTLs are ones where the effect is only observed in samples where you have a high enrichment or conversely a depletion of a given selected cell type that you're looking at. And you can see that on the right there. We can see an enrichment of a cell type. When that cell type is in low frequency, we do not see the EQTL effect. When it's enriched in the sample, you can see a genotype EQTL effect. And we did an extensive benchmarking to work out what the best approach for this was. There are no real best approaches, but we settled on using the Excel method. And what's shown in the left here is some of the cell types. And basically, gene expression was strongly correlated across many of the cell types. So what this meant was it limited our approach to looking at enrichment of a single dominant cell type for most of the tissues. What we did once we got these interaction QTLs was then to take 87 curated GWAS traits that were extensively curated in the Gtex paper, and to perform co-localization with these interaction QTLs in the same way we'd done it in the main Gtex paper for expression QTLs. And what we find here in this characterization in the top is a GWAS study from the UK Biobank. In the middle panel is the bulky QTL data, and we see no co-localization of this GWAS association with EQTLs in the bulk data. But down below is this interaction QTL data, and we see a very strong signal co-localizing. And we found for these traits that over 35% of these loci are co-localized only with an interaction QTL with these traits. In addition, we found a further 30% of loci that showed joint co-localization, where we had both an interaction QTL co-localized and a bulky QTL co-localized, indicating the cellular specificity of that trait and potential cellular origin as well. Furthermore, these cell type interaction QTLs are far more specific than the EQTLs in terms of tissue specificity. On the right, you see the high level of sharing of these standard EQTLs across the tissues. But these cell type interaction EQTLs, here looking at epithelial cells across these tissues, show far more tissue specificity. So these deconvolution approaches then revealed considerable cellular specificity across the Gtex data, but unfortunately they don't necessarily tell us the specific cell type in which the QTL is active, only that there's an interaction between cell type and the QTL. In addition, many more GWAS loci actually co-localize only with these cell type specific signals, and not with the bulk signal, indicating that this is really where we have to get to understand these disease associations. But to do this, we'll need to actually map QTLs in the individual cell types for these multiple tissues and organs to reveal these specific regulatory things. These deconvolution approaches only take us so far. And those studies have just started to be done. Obviously, I didn't have this morning's wonderful study. It isn't published yet. So all I can refer to is this study from Luda Franke's lab from last year, which was relatively small compared to this morning's study. But it too is from blood, so PBMCs from 45 donors. and they found unique eQTLs detected only in the single cell data. And some of the highlights of these type of studies are that you can see things that get easily masked in bulk data. So the top panel shows that you can identify a cell-specific weak signal that would be completely swamped in the bulk data. And the bottom shows that you can identify two cell types where you have these opposite effects that again would cancel themselves out, and you wouldn't be able to observe that in the bulk data. So how do we take that back to tissues? And that's one of the questions we started asking about a year or two back. And we wanted to know, can we go back to tissues that we've collected? Do we need to collect new tissues? Can we go back and use Gtex? If we looked across the Gtex tissues we had, and unfortunately many of these were preserved in a manner that wasn't amenable to current single cell techniques, and still aren't amenable to that. I would say that they're not going to be amenable, but this field changes so fast that it may be the case that in a year from now, all of those banked tissues may be amenable. But there were a set of tissues that we collect in Gtex that were freshly frozen, and those are amenable to single nucleus techniques. So we started to go across these tissues to apply those multiple approaches to determine which is the best approach and whether they work across the tissues. And what we ended up doing was to take all 8 frozen tissues from Gtex, to survey those across 3 individuals per tissue and to do 3 different single nucleus protocols each. So in total, we profiled about 300,000 nuclei. And the joint analysis of all of those across all of those tissues is shown on the right. Right now, we're in the process of looking at shared and specific cell types across these tissues to look at them in the context of the muscle tissues we have that span at least 3 of the tissues, and to look at them with GWAS colocalization analyses. We do hope to get this data out soon. I know several of us have showed it quite a lot recently, so we're making a push to get this paper wrapped up so we can get these data out fairly soon. But what we want to do is really to scale that up. 3 donors isn't enough. We really want to scale those studies up in tissues so that we can look at these QTLs that range from cell type frequency QTLs across the tissues to cell type specific expression, open chromatin, and many other molecular QTLs across those cell types. I'm just going to go back for a minute to some of these metadata and covariates that I was discussing and the importance of them. This is just looking at the lung data we generated for Gtex. And what we're showing here is several cell types that we annotated and genes that show very strongly specific expression for those cell types or strongly enriched expression in those cell types. And I've highlighted one here for the gene SFTPC in the alveolar type 2 cells. So that seems to be quite a specific marker for that cell type. And if we go back to our plot of all the Gtex lung tissues plotted by the first 2 PCs, we have all of the Gtex lung tissues here. And if we look, add on to that this marker, the expression of this marker across those Gtex lung tissues, we can see that it's not uniform across all the lung tissues. It shows far greater expression in this subset of lung tissues for these donors on the left here. And then if we go back to the covariates and metadata we had, you can see that that's almost an identical pattern related to the death classification scheme we used. And it turns out that that group clustered on the left is the Hardy scale 0 classification, corresponds to all of the individuals who were on ventilators. And this is something that was shown at the pathology level a couple of years earlier. What we see here is a normal lung on the left with a normal ratio of type 1 to type 2 pneumocytes. On the right is one with pneumocyte hyperplasia and in the alveolar walls. So we're getting more type 2 alveolar cells. And this is basically due to ventilator-associated lung injury, which is well known. So you can see it here in the Gtex tissues we were looking at with the a single nucleus sequence. We were only looking at these individuals with the type B, so they were only the ventilator individuals that we collected the frozen tissues on. So it's really important to have all of this data to untangle real biological signals from these associated covariates. So where are we and what do we need to do next? Obviously, we need eQTL data, or QTL data, I should say, at the single cell level. We need to pass out these cell type composition and to get expression, splicing, and other QTL and molecular phenotypes at the single cell level. We need to do that for multiple tissues. Many of the efforts obviously will have multiple tissues represented in the baseline map, but ideally we'd like to do it, also get it for multiple tissues within individuals so we can understand how that genetic effects affecting tissues within individuals. We clearly need different molecular phenotypes, and several talks have shown today how we can get more than one or multi-omic phenotypes. Particularly epigenetic phenotypes, again, across cells at the single cell level, so we can really start to understand the functional consequences of these genetic variants. And ideally, we need more samples. We need fairly large sample sizes for these studies, which I realize is a pretty tall order given the price tag of doing these studies. But to get to trans signals, we're going to need large sample sizes, and to get at weak effects, we're also going to need large sample sizes. Although that may be ameliorated a bit by actually having single-cell data where we can detect weak effects in the cell where they're active and not have that swamped by heterogeneous cell signals. And then finally, what we really need is population and geographic diversity. So Shyam showed this morning and basically set the tone for this. Gtex, like many other big genomic studies, is not very diverse. This is an old study. It doesn't have the complete set of donors, but it hasn't changed much. In essence, 86% of Gtex donors are white European in origin. We have only 12% African American, 1% Asian, 2% Hispanic. And this is something that's become sort of a growing cry in the genomics community. These are two studies that came out in 2016 and 2018, basically saying that genomics is really failing us in diversity. And this has ramifications for drug differences and for them to be effective and safe in different populations because of these different genetic differences. In the study in 2018, you can see on the left is the racial breakdown of participants in genome-wide studies. The vast majority are European. That bar graph on the right represents their, compared to the representation across the general population. So we're vastly European biased in these genomic studies relative to the population. And then these two studies have just come out this year, basically saying the same thing. And again, what they're showing here in this graph is that the percent of GWAS studies in the GWAS catalog is about 53% European, and the percent of individuals in the studies is about 78% European. So this last study on the right just came out, and that was actually a study sponsored by the called the PAGE study, which was a multi-ethnic group. So they did an analysis in that diverse population and concluded that effect size heterogeneity differs across the ancestry, and so it's very important. And we see substantial benefits for fine mapping and insights into clinical implications if we can study these more diverse populations. So it's something that's very important, and really in a nascent study like the nascent community and like the Human Cell Atlas, it's something we should address right off the bat going forward to make sure we have global, racial, ethnic diversity represented. So I just want to go back to Gtex and say that it wasn't, we did actually include and tried to include all the different racial groups in the US in the study, but part of the issue is related to consenting and different willingness to participate in studies. And we assessed this in the course of the study by running a standing ethics study that ran alongside the study and interviewed people who did participate and what their reasons were for participating, and those who didn't and what their reasons were, and tried to get at different groups that were underrepresented. In particular, they set out to approach the Hispanic community in the US to find out why these groups weren't participating in the studies and what it would take. These are a set of posters. I'll just show a few of them. On the left is the English, on the right was translated into Hispanic. These were larger than life-size posters. The ethics study basically enrolled a community advisory board from the Hispanics community and went into different communities at different times and basically discovered that you needed an outreach, you needed some education in the form of these posters and some film clips that they did. And really community engagement. And when they did that, they made a huge impression on the community and changed people's perceptions of biobanking and participating in these studies. So, as I say, there are several of these, but they had a big impact. So in summary, I think the Gtex project can provide both insights and tools for standardizing and optimizing approaches to tissue collections that are very pertinent to the Human Cell Atlas. But obviously, this is a distributed effort across many labs globally, and you can't really impose a Gtex-like structure and say that we can do that in many different locations. However, there are many lessons and pieces of Gtex that we can take and apply to what we're doing. So those post-mortem collection approaches and using post-mortem donors for obtaining normal tissue can be applied. If we can't do it across the body because we had the OPOs willing to do that in the US, there are still, as people have said today, as colleagues have said in Spain, there are organ transplant units and there are often surgical research surgeons who are participating in those who may be willing to apply some of these approaches to get a handful of tissues, even if it's not 45 or 54. Those SOPs that were created can serve as a baseline for how we might create similar SOPs for sampling tissues going forward. Or if we don't create similar SOPs, because some of us are doing biopsy sampling, some of us are doing surgical sampling, some of us are doing post-mortem sampling, maybe they can serve as a way in which we at least describe the sampling we did so that it's rigorous and other people can use it as a reference and that becomes part of the DCC. There are consents. I'm a member of the ethics working group. The Gtex consents have been used extensively as a baseline on which to help develop the consent modules and templates that the EWG are producing as part of their ethics toolkits. And metadata, there have been several, obviously I've harped on that a lot. The more metadata you have, the more you collect, the more valuable it is for downstream analyses. And so I've been working with the DCC in several fronts to engage that component. So the EQTL analysis across the tissues and individuals in Gtex was very powerful, and clearly it's a powerful approach to understanding regulatory variation and the sharing and specificity of that. But we need to map these QTLs at the single cell level, and that's really crucial going forward for novel colocalizations with disease and the cellular origins of these genetic disease-associated associations. And finally, what we really need is greater population diversity. We can't go forward and repeat what the genetic studies have done to date. We really need diversity and representation, and we need, at least at the level of the reference maps, to make sure we have that as a baseline. Even if that's not true for all of the GWAS or QTL studies going forward, because those will depend on where we can get very large sample size for a given tissue or for a given blood or for a given population. So I'm just going to finish there and thank the Gtex Consortium, who have been pretty active, and in particular the donors and the families who donated tissues to Gtex, and the various groups, including the RegEv group at the Broad, my own team, the Portal team, several from the analysis working group who helped generate some of the data and analyses. And I'll take a few questions, and Alistair can have his spear.
Wonderful overview. It's actually daunting to see how much work you guys put into doing all of this work, systematically collecting the tissues from the same site in each individual, characterizing them to see what confounding factors you might have, and then imposing this on a very, very large scale, harvesting so many tissues from one individual and doing that for so many individuals. I don't think our single cell efforts, at least not ours, are at that level yet in terms of scale and harmonization. But really learned a lot from your overview. The question is, so okay, one more quick comment is, it's also sobering to see how much variability you found in tissues across individuals. And you saw that because you had, I don't know, H&E stains or other kinds of ways of kind of visually looking at those tissues. Did you turn that into data that you could regress out as a confounding factor in the QTL analysis?
Yes, we used a peer analysis in the EQTL analysis so that we could, so we used a number of peer factors until we sort of maximized our EQTL study so we could regress out all of that. But we also went through systematically and looked at what those peer factors were most correlated with across the metadata we had. And, the metadata, as I say, goes back. We included everything from sampling times, detailed ischemic times, and it was detailed. They actually had to document when the donor died, when the tissue was taken out, when it was frozen, so very detailed clinical data, the time at which it was placed in one fixative and changed to another, and so on, and all the way down through till the batch effects people have been talking about today, where we have different individuals processing samples, sequencing batches, and those are huge as well. And, the tissue heterogeneity is a big one in our data, so it correlates a lot with, the top peer factors that we're regressing out. Ischemic time is another one. Gender and ethnicity was another one, and then all of the batch effects through to processing, sequencing, and those. So we could sort of correlate which ones were key, but many of them contributed in different ways. So in terms of the QTL analysis, we pulled a lot of those out. But for some of the different analyses we've been doing, they're not always removed in the same way that was discussed today in the immune cell group. So.
Great.
I'm not going to ask for the $5,000 beer, but I know the cell frequency QTLs. So when I was sitting watching Sounds talk, and I was thinking that data set would be perfect for it. But with the tissues, as you guys just mentioned about the sampling heterogeneity, that must make it much harder to actually do this in tissues rather than in bloods, where you can...
Exactly. Now, you could narrow it, if you have that wealth of data where you have an image on everything and a pathology review and you can see that, you could narrow it down to sets of tissues that look the same and do that. But clearly that's going to add noise, so the only way to get over noise is to do many, many more. Or to narrow it down to a set that's the same, but if you have that level of data on them, which includes images you can scan into, many of these samples are actually going to be highly amenable to some of the spatial techniques, because although they weren't preserved in a manner we could get nuclei out of them for single nucleus, they're beautifully preserved for any sort of histology and other spatial techniques. So we could define a smaller subset and try to look at those. But yes, you're right, that adds a level of noise. That was a nice ideal, but in these type of systems, there's a lot of noise. So,
cool.
Yeah.
Hello, very nice talk. We're from Singapore Institute for Clinical Sciences, and we have done for the Asian pregnant woman EQTL and mass QTL. So my question is, how can we contribute to the big map of the Gtex from how can we contribute from our data set and our result?
So you have...
we have done the EQTL for the whole blood, maternal blood for 238 samples, and mass QTL for 900 pregnant women whole blood. So for this result, we have now doing the manuscript. My question is, how can we contribute to Gtex, the big map?
That's a good question. We maintain a Gtex portal, and we actually have quite a few people who would like to have data added to that, and we've been very willing to do that. One of the things we'd like to do is to have everything run on the same genome builds and through the same pipeline, because it makes it more uniform. But we are considering, right now we're hosting some some data from eyes. We have missing tissues in Gtex, and I can't tell you how frequently somebody will email and say, "You're missing my favorite tissue. When are you going to do this?" And we have to say, "Well, I'm sorry, the funding stopped. We have what we have." But many individuals have said they'd like to contribute those data, and as long as it fits at least the basic data types, it's very easy in a sense to integrate it with the portal and to say where the study came from. So we're working right now to integrate a set of data that's from retinal samples from the eye, which includes expression from the retina as well as QTLs generated from those as well. So
Thank you.
A really quick question. I guess hindsight is always 20/20. So if you had to do one thing differently for going forward, what would that be in terms of organizing such a big effort?
I'd make it go slightly larger.
Okay.
And I would collect frozen samples for everything. There's no way you can do a study like Gtex with fresh tissues. You simply can't have somebody sitting there in the operating room for 54 different tissues as they come out and make it work. But you can, how do we preserve them best in a way that's going to have maximal use for different studies? And it turns out that there was sort of pilot studies done at the time, and we used a sort of fixative for many of the tissues that was very superior for RNA quality and also superior, much better than formalin fixation for histology as well. So it was great for some of the purposes at that time, but it didn't think forward to some of the other needs. So those tissues are actually not really amenable to some of the histone studies where you're trying to get at the DNA-bound proteins, and they're not really amenable to trying to separate nuclei out for those. So I would have collected more tissue and preserved it in a manner that we think might be more usable. Because this entire data set we could go back to if that had been the case, and it's not the case. So.
Thank you. Yeah.
Okay, so I think that's about time. Oh, you have one more? Okay, one more.
Just a quick question. If I understand correctly, you're saying 900 was not enough? You said if you do it again, you'd do it bigger?
Yeah, we're only just starting to get to trans, and trans is interesting. We're finding some trans signals, strong trans effects. They're rare, but when they're there, they're very interesting. So you need, and to detect the small effect sizes, which we know we're going to get small effect sizes. We have small sort of association. We have small effect sizes in GWAS studies. So to really estimate those, we need larger sample sizes. But in actual fact, we could do, I wouldn't necessarily continue Gtex doing the way it was doing, but I would probably maximize some of the, getting larger sample sizes across different ethnic groups so we can understand the differences, but you can also bring those together, account for that, and look across the large study. So we could get the large and the diversity at once.
Thank you.